Hello everyone, my name is Phan. I'm a first year graduate student in the Bioinformatics and Computational Biology program at Iowa State University. I'm currently in Dr. Kadelka's lab and Dr. Ido Friedberg's lab. Today, I will walk you through how function annotation inequalities reveal the need to study under-annotated genes. We biologists have been trying our best to predict functions of all identified proteins. Some proteins are very well studied, leaving many others with little information known, called the ignorome. Within this ignorome, some proteins are currently understudied. They may have valuable information as missing pieces to the bigger picture. We used gene ontology annotations from 2011 to 2020 to examine the gap in knowledge between the highly annotated and the under-annotated proteins, and eventually uncover some hidden gems in the ignorome. We start with gene ontology raw annotations from 10 years, then we use Go Thresher, a tool that we built to filter Go annotations. We select experimental annotations in human and use this threshold of 100 proteins to divide into articles describing high-throughput experiments or low-throughput experiments. For each protein, we have these data. Let's take A1BG for example. We can have some per protein metrics. First, the annotation count. Here is 5. Second, total information content. Here we have 4 unique Go terms, so the total information content would be a sum over 4 unique Go terms, where the PI is the frequency of a Go term in the corpus. Third, an article count. Here we have 5. We then have an O proteome metric, that is Gini index, which quantifies the distribution inequality of these 3 metrics in the proteome. I have some results I want to show. This is a box plot showing median and interquartile range, and the outliers are not displayed here. You can see that the proteins from high throughput experiments, the blue boxes, have low annotation count on the left and total information content on the right. So even though high throughput articles annotate a lot of proteins, they actually provide less number of Go terms and less informative Go terms compared to the low throughput experiments, the orange boxes. Also, the median total information content per protein from low throughput articles increases slightly from 2011 to 2020, indicating an information gain in the protein function space. Next, I have a plot of total information content versus annotation count of all proteins, where each dot represents a protein at one time point. On the top, we can see some proteins of high interest are heavily annotated. Let's look at Park7, where I picked out 3 time points describing its high growth in both annotation count and in total information content over time. For the remaining 5 proteins labeled here, there could be a saturation effect where the total information content is not linearly increasing with annotation count. Also, the low throughput annotations are exponentially more informative than the high throughput annotations, and because we see a lot of orange dots on the top. Besides that, we select the proteins annotated with only protein binding term. Over time, more and more proteins are repeatedly annotated with this low information term, which is always below 1, making up a part of the ignorome. Because of the big gap between the highly annotated and other genes, we calculate the inequalities of the 3 metrics. Gini index ranging from 0 to 1 is a measure of economic inequality that we adopted here. Higher Gini value will indicate a higher inequality. So we calculate Gini index for each of the 3 Go aspects over time. We can see here high inequalities in all metrics across all aspects suggests that we repeatedly study the already annotated proteins, which worsen the gap in knowledge. Also, if you look at the red line of molecular function aspect, we accumulate annotations more strongly in counts for the highly annotated proteins in the left figure, but there is no corresponding increase in the information content or the depth of knowledge of these proteins in the middle figure. Lastly, we have some co-expressed pairs of genes. The arrow points to green genes with high total information content, while we have almost no information for the red genes down here. We can see that co-expression reveals some members of the ignorome that are associated with highly annotated genes, but are not currently receiving much experimental attention. Therefore, it is important to study these under-annotated genes. In summary, articles describing high-throughput experiments provide annotations with low information content and also provide little additional information over time. More information is provided by articles describing low-throughput experiments. Secondly, high inequalities in the matrix distribution indicate that we repeatedly study some proteins that are already annotated. Some of the most highly annotated proteins are possibly saturated in information. And third, association with the highly annotated proteins can help detect understudied members in the ignorome. Here are my 3 advisors and lab members who help me with my work, and that's all I have for my presentation. Thank you for listening.